Precision Study is actually a pretty incredible research initiative, a novel approach to actually studying pulmonary fibrosis. The initiative is a collaboration between the Pulmonary Fibrosis Foundation. It's supported with a $22 million grant from the NIH and is also supported from a philanthropic organization, Three Lake Partners. What the study does is it allows us to take some of our knowledge about the genetics and our deeper understanding of idiopathic pulmonary fibrosis and really try to come up with targeted treatments for IPF patients. So based on some prior analysis, it was found that patients with a specific genetic variant may have a better response to therapy with N-acetylcysteine. So the first aim of this particular study is to actually screen patients, identify patients with this particular genetic variant, and give them N-acetylcysteine to see if there are differences in clinical outcomes. Other parts of the study will allow the identification of other genetic variants, some blood banking as well, to really use novel techniques such as transcriptomics and proteomics to really deepen our understanding of the disease process.
This is the first pharmacogenomic study that's ever been undertaken in the treatment of idiopathic pulmonary fibrosis. So we learned from the PANTHER study, which was a study of azathioprine, prednisone, and N-acetylcysteine, that there are subgroups of individuals that have differing responses to N-acetylcysteine. There's a subgroup that appears to benefit from it, and there's a subgroup that appears to be harmed by it. So a genetic polymorphism in the TALP gene, which stands for toll-like interacting protein, has been discovered that seems to correlate with the outcome in the effect of N-acetylcysteine in these individuals. So N-acetylcysteine is an orally available nutraceutical, which is metabolized in the body to glutathione. Glutathione is a very important extracellular antioxidant, which is known to be completely depleted in the epithelial lining fluid of individuals who have idiopathic pulmonary fibrosis. So the idea, the concept behind the use of N-acetylcysteine is to supplement a naturally occurring antioxidant in an organ where it's known to be deficient and where oxidative damage to the lung is thought to be an important mechanism in driving the pathology that leads to pulmonary fibrosis. So this is a very large undertaking because only about 25% of the general population and individuals with IPF have the genetic polymorphism that indicates that they'll have suggests that they'll have a favorable response to N-acetylcysteine. So in order to identify enough individuals for this clinical trial, many hundreds of individuals will have to undergo genotyping to see which polymorphism of the TALP gene they have. And the Pulmonary Fibrosis Foundation registry will be the initial source of subjects for this multi-center study. After individuals are identified with this polymorphism, then they will be randomized in a double-blinded fashion to receive N-acetylcysteine or placebo. The dose and the preparation of the N-acetylcysteine will be exactly the same as the dose and preparation that we used in the PANTHER study, so that we expect to obtain similar results. And we hope to show that individuals who have this favorable polymorphism in the TALP gene will benefit from the use of N-acetylcysteine while avoiding harming individuals that have the that have the unfavorable polymorphism. So this is a very exciting study. It's the first time that a pharmacogenomic approach is being used in the treatment of idiopathic pulmonary fibrosis. And we're really excited to get the study started.